My lab studies many aspects of RNA biology, but one of the things that we've done is try to apply our insights to develop a technology. In particular, what our goal is to use different RNA designs to target pest species. And what this does is it allows a completely biocompatible molecule that's very environmentally consumer friendly, so that you can eliminate a pest without any of the drawbacks of toxic pesticides or other things that might, be bad for consumers or the environment.
This project is really exciting because it's not a far-fetched future maybe. It's actually probably going to come to fruition even sooner than my lifetime, I'm hoping. We're working on using an understanding of the defense system that occurs in all living organisms, the innate defense system. And it evolved to recognize virus infections in organisms. And it recognizes viruses by noticing that these molecules are double-stranded RNA. Organisms don't naturally make double-stranded RNA. They make all other sizes and shapes and kinds of DNA and RNA, but not double-stranded. So the signal when an organism sees double-stranded RNA is, this is virus. And the innate immune system responds to that invasion, so to speak. The sequences are also recognized as foreign, not just the shape, but the sequences can be as well. And so the system is triggered to actually find and through a series of manipulations, eventually clip or cut or cleave the RNA, and basically disallowing the virus from infecting the plant.
RNA interference is a technical term, actually, that was established decades ago as a method to do gene silencing. It exploits a particular way cells process RNAs and then recognize things to be silenced. By using RNA interference, we should be able to introduce a sequence that targets an essential gene within that pest, which would kill it. Now, the useful thing about this is that the genes in the pest are very different from the crop, the humans that might consume it, or even other insects in the field. And so those will not be affected by that very specific sequence.
We would like to be able to develop biopesticides that would replace the harsh chemistries that are economically damaging, those that are harmful to people, those that are harmful to everything, and try to make them extremely specific and soft, so to speak. So we can control the specific insect that we would like to control over all others.
We received some funding through the Mississippi Smart Business Accelerator Initiative, which allowed us to apply for an NSF ART grant, which is Accelerating Research Translation Grant from the National Science Foundation. This is a large multi-million-dollar grant to Southern Miss. Part of that is an association with a mentor institute. And in this case, it's the University of Arizona.
Alex Flynt had a graduate student, wanted to take the fundamental knowledge he learned in Alex's lab and apply it to solving an agricultural problem. So he contacted me, and I did have a project that was funded for 7 years, actually, to work on RNAi, or RNA interference as it's called, in an insect called psyllid. We're also working on whitefly as a very important vector of plant viruses. They all transmit pathogens to plants, and they feed in the vascular system of the plant, so any pathogen that's present there is a target for this technology. So Mosharraf came to the lab to work as a postdoc on the project, and we thought, well, this would be a good opportunity for this NSF ART grant. If food can be produced without harsh pesticides, and if we can make a contribution to reducing those harmful effects and improving the health of the environment and the organisms within it, as well as within ourselves, I think those are pretty heavy-duty challenges and goals.